Good morning, good afternoon, good evening, and welcome to day 2 of our program that will cover a range of topics around gene bank management and seeds in culture and society. I'm Sally Norton. I lead the Australian Grains Gene Bank. Firstly, I'd like to acknowledge the traditional owners of the land and pay my respects to the elders past, present and future, and extend that respect to Aboriginal and Torres Strait Islander people who are present today. Before we launch into our presentations today and our keynotes for the sessions, we have some housekeeping. First up, Albert Voss's presentation from yesterday that had some technical issues has been moved to this afternoon between 3:30 and 4:40 p.m. and his video will be available on the platform on demand under yesterday's session. A reminder that for any social media, please use the @SeedScience21 tag and tag the Seed Partnership as well. And don't forget to check out the on-demand videos available on the portal. And a reminder that on GatherTown today, we have a poster session between 1:30 and 2 o'clock. You should have links to GatherTown in some of the emails you've received. And again, this evening at 6 p.m. for a seed trivia night. So that'll be a heap of fun. So make sure you come along in there. Links to GatherTown will be in the portal banner as well, and the password is germination. Reminder also that links to those who've registered for the workshops will be sent out by email by the end of the day. And for portal assistance, please use the live support function or contact the email below. We also want to do a great shout out to Sue Everingham for organising the networking event last night. It was a fantastic event that was well attended. So thanks for that. And just an acknowledgement of all of our sponsors and partners there. Without all of these partners and sponsors, this event would not be able to run. So it's an absolutely amazing day yesterday, and today will be no different. And with that, I'd like to introduce Dr. Emilia Martin. She's currently a project manager for the Australian Network for Plant Conservation and has coordinated updates for the third edition of the Plant Germplasm Conservation in Australia. And I think that's a bit like herding cats, so I think Amelia has done an amazing job. Amelia has a research background investigating seed lifespan and germination with a focus on improving seed quality for conservation, storage of Australian native plant species. She has also a growing interest in science communication. Amelia's research is based at the Australian Plant Bank, part of the Australian Institute of Botanical Science. When she's not at work, Amelia is usually looking after her time-intensive germination experiments, namely her 3 kids, or reading a good book. Over to you, Amelia.
Thank you, Sal. I'm proud and humbled today to join you for the launch of the 3rd edition of Plant Germplasm Conservation in Australia. When Professor Lesley Hughes launched the previous edition in Melbourne in 2009, she noted that these guidelines were a message of hope, and I've looked at her inscription many times while we've been working on this new edition. I'd like to suggest that this 3rd edition is a message of hope and action as we come to terms with the urgent need to conserve plant diversity, while the threats are overlapping and increasing in magnitude. As humans, we desperately need to see hope combined with action. We need to look out for projects, initiatives, and individuals who are working so hard to conserve plant diversity, habitats, and threatened species, and to use ecological restoration and translocation to win back landscapes. The Australian Network for Plant Conservation is proud to have played a part in sharing knowledge and connecting people and organisations dedicated to conservation of Australia's native plant species over the last 30 years. Production of this handbook for ex situ plant conservation has been an incredible team effort. The lead authors, co-authors, case study contributors and steering committee have revised 8 chapters and drafted 7 new chapters over the last 18 months. I'd particularly like to thank the lead authors, Cathy, Marlene, Anne, Dave, Karen, Meg, Bryn, Amanda, Nushka, Tom, Danny, Graham, and Andrew. Their co-authors each brought something important to the table, and each writing team was responsive and persistent through the revision process. The first 6 chapters provide a step-by-step guide to germplasm conservation, starting with planning, followed by genetic guidelines, collecting strategies, seed storage, and germination. The next 4 chapters discuss techniques that may complement seed storage or be the only mechanism of exceptional species, such as tissue culture, cryopreservation, nursery practice, and living collections. The following 2 chapters outline the types of germplasm collections suitable for species with special requirements, such as orchids, fungal symbionts, ferns, carnivorous, and parasitic plants. The final 2 chapters provide practical advice for managing risk within collections and strategies for responding to crisis situations, as well as guidelines for the maintenance of use of all types of germplasm collections and associated data. Please show the chapter authors and co-authors your appreciation in the discussion. This edition includes an incredible 50 case studies that highlight the aims, processes, and partnerships that underpin ex situ plant conservation. I encourage you to read every one, to learn from what these authors are sharing, and let yourself be inspired by their actions. Whether they are encouraging volunteers to get involved, refining techniques for breaking dormancy, bulking up collections through seed production, or using collections for translocation, there is so much we can do to make the best use of every germplasm collection. Please show the case study authors your appreciation in the discussion. We have dedicated this edition with gratitude to all those who introduced us to the beauty and complexity of Australia's flora. We owe a huge debt of gratitude to our families, friends, and colleagues, past and present. They are also dedicated to the children and grandchildren we know, who inspire our efforts to conserve Australia's biodiversity. The last 2 years have reinforced that the relationship we have with the natural world sustains us in so many ways, and the relationships we have with each other make life meaningful. On that note, I would particularly like to thank the Germplasm Guidelines Steering Committee for their support. Joe Lynch, Lucy Commander, Cathy Offord, Bob Makinson, David Coates, Linda Broadhurst, Damien Wrigley, Sal Norton, Kath Whitehouse, Lydia Guza, Neville Walsh, David Bush, Rebecca Stanley, and Tony Alls. I also owe a huge thank you to my colleagues at the Australian Institute of Botanical Science. It is a pleasure and an inspiration to work with people who love plants so much. This work would not be possible without the support of our generous funding partner, the Ian Potter Foundation, and our project manager there, Louise Arkles. Their funding allowed for project coordination based on attainable and flexible goals. Importantly, it also means these guidelines and accompanying videos are beautifully presented and available for free download, which is critical for accessible and open sharing of this valuable information. I'd like to introduce the CEO of the Ian Potter Foundation, Craig Connelly, to say a few words ahead of the official launch. Craig commenced as CEO of the Ian Potter Foundation in 2015. He spent 25 years working in a variety of roles in the Australian financial services sector and is now dedicated to contributing to the community sector. Craig is an experienced business analyst and successful manager and has been an effective mentor to many young professionals throughout his business career. Craig has previously been an external member of the Advisory Committee for the Sustainable Farms Initiative at ANU and a member of the Natural Capital Expert Advisory Group. He's founding trustee of the National Parks Conservation Trust and currently a director of Water Trust Australia and SANE Australia. Thank you for joining us today, Craig.
To you, and good morning everybody attending from wherever you are located around Australia this morning and internationally. I'd also like to acknowledge that I'm coming to you today from the traditional lands of the Wurundjeri people of the Kulin Nation, and I pay my respects to elders past, present and emerging. The Ian Potter Foundation is very pleased to have supported the Australian Network for Plant Conservation with a $200,000 grant to comprehensively update their flagship publication, Plant Germplasm Conservation in Australia, Strategies and Guidelines for Developing, Managing and Utilising Ex Situ Collections. That's a mouthful. In preparing for this session, I chatted with Amelia early last week, and throughout that conversation, it became obvious that there are layers of positive outcomes that have flowed from this relatively straightforward and modest grant from Potter's perspective. It's clear in Amelia's opening comments that the steering committee has been incredibly influential in overseeing the update of the guidelines. They've leveraged significant in-kind and expert support from eminent individuals and organisations who've all contributed immensely to the content review and update of the guidelines and have done so pro bono. The fact that such support, coupled with the efforts of members of the Australian Network for Plant Conservation, means there's a strong scientific basis for all updates to the guidelines as now published. That's critical to Potter. We've been a strong supporter of scientific endeavour since the foundation was established in 1964. The funding also supported efforts by the Secretariat to engage leading scientists at different stages of their careers and from a geographically dispersed and diverse area to contribute to the update in an inclusive and engaged way. And pleasingly for IPF, and as Amelia has already articulated, the dissemination of the updated guidelines is going to be to a very wide audience and is free. And possibly an unintended consequence of the funding that's been provided has been to facilitate two-way exchange of material with international contributors, effectively raising awareness of the updated guidelines and also facilitating connections that might foster future collaborations. As a funder that's supportive of scientific endeavour, this project ticked a lot of boxes for Potter. The work ensures that the guidelines remain relevant, they're up to date, they're available to as wide and an engaged audience as possible, and they're based on world-class science. And a small budget has also been set aside to evaluate the project, and I look forward to discovering the evaluators' conclusions in years ahead. It's now my pleasure to introduce our next speaker, Dr. Cathy Offord. Dr. Offord is the Manager of Germplasm Conservation and Horticultural Research for the Royal Botanic Gardens and Domain Trust. She is based at the Australian Plant Bank, part of the Australian Institute of Botanical Science in southwest Sydney. Dr. Offord is also an adjunct professor at the School of Biological, Earth and Environmental Sciences at the University of New South Wales. Specialising in the conservation and horticultural development of Australian plants, Cathy's work encompasses seed biology, tissue culture, cultivation and translocation of threatened species. Cathy was the lead editor and author of several chapters of the previous edition of Germplasm Guidelines and the chair of the steering committee for the revision and publication of the third edition. Welcome, Cathy.
On behalf of the steering committee for the third edition of the Australian Germplasm Conservation Guidelines, I want to sincerely thank all of the contributors to this and to the two previous versions. As I reflect on my own career as a conservation scientist, I can measure the real-time increase in ex situ conservation science by the size and the scope of the successive guidelines that have been produced. The first guidelines were published by ANPC in 1997. These were groundbreaking guidelines and they ran 40 pages, which were overseen by the editors Darren Tushel, Mark Richardson and Kingsley Dixon. This document became the wayfinding document for those of us who were grappling with the burgeoning needs that emerged in the 1990s to ramp up scientific efforts to conserve plants ex situ and in situ. 12 years later, in 2009, Trish Marr and I edited the second edition, and the scope and the magnitude of the information that was available had increased to the edition to 190 pages, including 19 authors. Fast forwarding another 12 years brings us to this 2021 revision. This is a compendium of great science and practical wisdom. It reflects the vastly expanded activity in science and practice of ex situ conservation over the past 12 years. In all, there are 15 chapters and an astounding 50 case studies written by 78 authors. Added 5 reviewers, and there are more than 330 infographics, illustrations or graphs. The printed document itself will run to an impressive 500 pages. This latest version has been overseen by the extraordinary Emilia Martyn-Yenson, who is herself a researcher and contributor to Australian plant science and science communication over many years. Amelia has wrangled the multitude of authors and reviewers with gracious tenacity, and it's due to her efforts that we are launching these guidelines on time and on budget today. I also want to mention the incredible Siobhan Duffy, who's made these guidelines a pleasure to read with her amazing design and editing talents. The constant in the years between the successive versions of the guidelines is the never-failing support of the Australian Network for Plant Conservation, who, despite often lean times, have always been there to ensure collaboration between scientists and conservation practitioners, and to provide workshops, conferences, guidelines, and other products to enable active plant conservation. Thank you, Joe Lynch and the team at AMPC for providing the knowledge bridge between science and effective conservation policy and action. I must also mention the support of the Australian Seed Bank Partnership, the powerhouse of Australian seed conservation. Thank you to Damien Wrigley and to all of the other seed bank partners. I'm proud to have been involved with the development of these guidelines, and I'd like to acknowledge the wonderful support and thoughtful guidance of fellow members of the steering committee. Special thanks goes to Linda Broadhurst for heading up the application for funding of these guidelines, which has been provided by the Ian Potter Foundation, who have been so supportive in our collective efforts to and it is now my great pleasure to introduce Professor Tim Entwisle to officially launch the 3rd edition of the Australian Germplasm. He is a highly respected scientist, scientific communicator and Botanic Gardens Director. Tim is currently the Director and Chief Executive of the Royal Botanic Gardens in Director of the Royal Botanic Gardens and Domain Trust in Sydney. Tim is an expert in freshwater algae, but he has a broad interest in plants. Tim.
Thank you very much, Kath, and it's great to be here today and lovely to be invited. I'd first of all like to acknowledge that I'm speaking also from Wurundjeri land and pay my respects to elders past and present, particularly any elders who might be with us here today. This is an amazing occasion, and I was thinking when I was preparing for this today that we're often very apologetic about seed banking and in fact about ex situ conservation in general. I suppose we're very aware that recreating nature from a few packets of seed is close to an impossible task. That's kind of not really what it's about. But the fact that we're already making withdrawals from our germplasm banks for restoration, relocation, responding to bushfires, climate change, that relentless land clearing, demonstrates, as if we needed it, that the situation is quite dire for our native biodiversity and how important things like ex situ conservation are already and are becoming so. So while we know its limitations, seed banking is important, but not only that, it's quite romantic and very uplifting for many. And I don't think we should lose sight of that. I think everyone has collected or stored or tried to germinate seed, and that can be a tomato plant, it could be an avocado, or it might be a local gum tree. And seeds are often metaphors in songs and writing and used for hope and renewal, from little things, big things grow, and many such lines. And for those of you who might be wondering about the bad seeds, apparently that band name comes from a 1956 horror movie, so it's entirely unrelated to all this. But look, seeds are fascinating, and old seeds are even more fascinating. 16 years ago, I was thinking, when I was freshly appointed as director at Sydney's Botanic Gardens, and there was a media report came out about successful germination of a 2,000-year-old date palm. Now, very few seeds last more than a couple of hundred years, and even that seems a little bit excessive in terms of evolutionary advantage, but there you go. When I was preparing to talk about the old palm seed on Sydney radio at the time, I of course chatted to Cathy, who's just introduced me, and did a bit of Google research, as you do. And people repeat stories about wheat seed surviving in the pyramids and Egyptian tombs, but these are mostly carbonised, non-viable seed that they might look okay, but they're dead effectively. There's records of 10,000-year-old or perhaps older lupin seeds found in Arctic lemming burrows. These are also dead. And in fact, I've got an acorn on my bedroom windowsill at the moment that's 12 months old, and that's probably dead as well. I only found out recently that acorns only live for about 6 months or so, even in squirrel hollows. So I'm going to just keep that there for beauty purposes only on my windowsill. But with the right care and conditions, seeds do last for a long time. And prior to that 2,000-year-old date palm seed, the oldest known seeds to germinate were lotus seeds, I think, a Nelumbo from a lake bed in China. These were said to be 1,288 years old, so very old. And they did germinate, but didn't quite make it into fully grown plants, which I do remember is something that Cathy was explaining to me, that seeds can be alive, but just alive, not quite enough. And there are successful seedlings that have been grown from 400, 450-year-old lotus seeds, although they were still a little bit wonky too, I think. But, you know, when you get into the hundreds, there are convincing stories, and there are, you know, there's a story of a 600-year-old seed from a ceremonial rattle in Argentina. Q always tells its story about the 200-year-old leucospermum seed from Africa, which was stored in a leather wallet and could be, you know, regenerated and grown into a plant. In Sydney, we had our own tale from 1890 when there's 100-year-old golden wattle seeds that have been stored in the belongings of someone's deceased mother. And they were germinated, and 8 out of 10 of those survived 100 years. So these are great sort of headline-grabbing stories, but they're not really, as everyone here would know, what seed banking is really about. A couple of wheat, lupin, lotus or date seeds is not going to protect the world from rampant land clearing, extreme bushfires, pests, disease attack, not to mention climate change. So that requires something altogether more sophisticated than a wallet or a dead person's chattels or even a pyramid, let me say. We need the infrastructure, we need the seed banks, the nurseries, we need the knowledge, and we need that know-how about what we're doing, which is why I'm delighted to be here today to help launch these guidelines, partly because I do enjoy a good story about old seeds, but I'm also aware that seed banking is much more than that and quite complicated and complex. Now, I know that because I'm, as you heard, head of Botanic Garden here in Victoria, which is one of Australia's 10 conservation seed banks, as well as we have here a flourishing orchid conservation program. We have a spore bank about to be created, and a recently expanded ex situ living collection program in our nurseries too. I was previously head of the Botanic Gardens in Sydney when Plant Bank was just a twinkle in a few people's eyes. We had a promise of a few $1 million to build it, and the money came a year after, I think, the launch of the second edition of these guidelines in 2009, which was also, interestingly, the year we sent some packets of seed to the space station with an astronaut, Greg Chemetov. Speaking of stories that add to romance more than science. But anyway, it was a fun time back there in 2009. And then after that, also, I spent some time at Kew Gardens for a few years looking after conservation living collections and estates there, which gave me responsibility for the wonderful Millennium Seed Bank. And that, of course, is a founding partner for so many seed banks around the world. I'm also delighted because I'm a member of the Council of Heads of Australian Botanic Gardens. As you've been hearing, they're governing or looking after the Seed Bank Partnership in Australia, and which is our premier germplasm network, of course. And working in a botanic garden does give you a particular perspective on all this. And I'm not going to talk today about the value of preserved plants in our herbaria, the learning programs, the activities that we do to encourage changes in behaviour, or about scientific research that goes along with those preserved and living collections. There's a whole lot of things that botanic gardens do, of course, that contribute to conservation, contribute to fighting climate change, and are associated with seed banking. But I did want to say a few words about what I like to call conservation horticulture. If seed banking is not just about old seeds or sending seeds into space, what is it about? Well, I think it's about horticulture in its broadest sense of that term. And I've been fond of saying recently that gardening is the answer to our plant biodiversity crisis. It's an oversimplification, but I do think that gardening is going to be a key part of what we have to do with the kind of impacts that are happening, particularly with climate change, but also big things like bushfires that are driven by climate change. Now, conservation gardening, or more formally conservation horticulture, also used to be a bit of a fringe science, perhaps a little bit like seed banking, and it was lumped in as something at the side of kind of where real conservation took place. And as I said at the very start, we now know that's not enough. You know, we need to draw on our seed banks now. And in a crisis such as our response to climate change, we do need to base our action on the very best scientific evidence and very best advice. And that all points, I think, to a very well managed ex situ plant collection. Now, none of us want a kind of a parallel universe of living dead plants in nurseries and fridges while the plant steams and burns and drowns. That's not the outcome we want. But just as we accept hospitals, care units, medical intervention for humans, particularly at this time during the pandemic, we have to embrace, I think, ex situ conservation in that same vein. While the analogy with medicine does have a few weaknesses, another strong parallel for me is that these ex situ conservations are partly insurance, but they're also for research and other purposes as well. So it's for healing now, as well as gathering the knowledge we need to heal later. What I like to call a plant rescue and care unit, which is like an intensive care unit in a hospital, is now really part of our business here at Royal Botanic Gardens Victoria, and part, I know, of the business of many of you here today. So I was very pleased to see in this new guidelines a section on propagation and nurseries being pumped up a little bit. So some of the chapters here have been really added to since the last editions. And as with so much of the book, drawing on expertise from all around the country. I think that's a real strength of these guidelines, the many voices, the many areas of expertise, and the fact they draw it all in from all those different places. Now, the problem we are addressing is, of course, a rather big problem. There's 330,000 known plant species alive today that produce seeds, or named plants. We do know there's a lot of seed banks in the world. That's been calculated. There's 1,750 of them in the world with about 6 million accessions, but, and the but is, most of these hold crop plants and wild relatives. So it's the conservation seed banks we're really interested in here today. There are about 50,000 species or taxa in conservation-based seed banks. And of course, thanks to orchids and things like that, there's millions of seeds as well. But 50,000 species in those seed banks, possibly more. And that means that we've got at least 15% of what we know of the world's seed-bearing plants already stored in seed banks. That's a really positive achievement, I think, actually, to be on the way to holding that many seeds, and probably well beyond that now. Importantly, of course, many of those are with a single accession or collection, as again, it comes out through these guidelines, it's critical to have more than that. Here in Australia, with about 23,000 vascular plant species and 84% of them not growing anywhere else on the planet. So this is an amazing flora we're looking after. If I interpret that, what I read correctly in the guidelines, our Australian seed banks hold perhaps 10,000 of those, and that's in about 20,000 separate accessions, with most of them, I should say, duplicated in the Millennium seed bank at Kew. So we may have nearly half of our known species, Australian plant species, in seed banks, although I do know that figure's a little bit rubbery. I know Damien is working on getting a real estimate very soon on that. But again, a handful of seeds for each species, and on average, perhaps it's about 2 collections for each. So there's a little bit of work to do there, but we are starting to get that material into the bank, ready to pull out when we need it for whatever purpose. Our focus at the moment, though, of course, is on rare and threatened species, and about 10% of Australian plant species are threatened with extinction, or we know are threatened with extinction, and many more are rare, and I would suggest on the edge of listing as well. So what we do know more precisely, and you can read this in the guidelines, is that we have close to 68% of federally listed species, that's about 930 of them, in our seed banks. Now that may drop a tiny bit, or who knows how much, as we discover what we may have lost after the recent bushfires, and of course will lose through climate change. So that number of endangered and threatened species is likely to increase, and we need to keep continuing to get more of those into the seed banks. Then, of course, there's bryophytes, there's fungi, there's algae, there's other things we might want to also store as well. So there is plenty of work to do, and plenty of work to do properly. But as Amelia has said today, this guide should definitely be seen as a message of hope. We can do something constructive to save plants, and this tells us how we can do that. It's evidence-based, it's science-based, it's best practice with 50 constructive case studies on how to care for seeds, for plant tissues and whole plants off-site and look after them when they're not in nature. I do sometimes get the feeling when I talk to people outside this virtual room that seed banking is like shoving a bag of seed into an old cupboard, or or a pyramid, or preferably most people think under a few meters of permafrost, but then shutting that door and walking away. And then suddenly you can magic back the habitat by going in, pulling the seed out and popping it back in the ground. Now, anyone assisting with the incredible community restoration, revegetation projects that are going on, anyone associated with seed banking, germplasm banking, will know that is not the case. It's a rather difficult thing to do and requires a lot of expertise and a lot of experience. And the difficulty and the challenge starts with the collecting and storage of that germplasm in the first place, which is why this collection of very wise words and experience from over 78 experts in the field is so important. There's really a lot in here. It's 500 pages and written by so many different authors and experts. There's advice on collecting, on storing and growing plants that you've heard, and then to be used for translocation, reintroduction and rewilding, or for research or public programs or other particular uses. It answers those kind of immediate questions. What do you put in your ex situ collection? How much of it? For how long? Which bits of the plants? And through detail on different storage techniques, and right through to breaking dormancy. And in this new edition particularly, there's even more information, as you'd expect, on genetic integrity, on difficult to store and grow plants. There's more on orchids, of course, special collections like carnivorous and parasitic plants, on spores, pollen, risk management, a whole lot more. So lots and lots of information. What does appeal to me, though, is the mix of practical and theoretical. So you can dip into this guide to whatever level you think is appropriate. And it ranges from beautiful graphics and lovely summaries through to tantalizing mathematical formulae and quite complicated experimental results. But it's really a little bit there, a little something for everyone. We're told that there's 10 conservation seed banks in Australia, including the one here in Melbourne, and holding, as I said, 68% already of our threatened flora. And I think in a positive vein too, 92% of Australian plant species are relatively easy to store in one of those banks. That is, they're not recalcitrant or non-orthodox, non-orthodox. Of course, as the book says, we want more genetic diversity in there. We want more plants, and we want more of those things that we haven't quite got into those banks already. But it is really good news. So I today want to finish by thanking everyone who's been involved in this production, congratulating everyone, to the authors, to the experts involved in this edition, and to those who were involved in the first two as well, because it does build on their shoulders. It really is a collaborative effort, and I can't imagine the coordinating and cajoling needed to bring it to completion. I gather from Amelia it's a bit of a COVID project, and amazing to do it during the last couple of years and get it to completion. So well done to all the authors, led by Amelia, Cathy, Trish, and the others that have been mentioned already. Friends, colleagues, and wonderful people. So they've done a fantastic job. So well done, you guys. And finally, I would like to thank also Ian Potter Foundation for showing their support. It's really good to see them supporting this project, and the Australian Network for Plant Conservation for their persistence and dedication to this cause. Now, just before I do the official launch, and I have to work out how to do this with a virtual publication, I'll think of a way. We're going to show and share with you a short video now that hopefully doesn't say all that I've said, but it will tell you a little bit more and in a far prettier way. Thank you.
from deserts to wetlands, rainforest, woodland, and alpine ecosystems. Australia's flora has evolved to survive diverse climates and stresses.
We have more than 23,000 plant species.
90% of the plants in Australia are only found in Australia. They're endemic to Australia.
24 years ago, the Germplasm guidelines were developed. These guidelines bring together our expertise in preserving Australia's unique flora and their genetic material away from their natural habitats. But in the same 24 years since these guidelines were developed, Australia's flora has faced increasingly complex challenges.
Australia has one of the worst global extinction records. The estimates of species extinction are between about 55 and 70 plant species, and more than 10% are still threatened with extinction. This is largely because of many co-occurring threats, including weeds, pests and diseases, overgrazing, habitat loss, changed fire regimes, and of course, climate change.
Some of the threats are so big, like a warming climate and the uncomfortable nature of how that feels. And the idea of having frequent severe wildfires covering most of eastern and western Australia, that's a serious issue for everybody who lives in this country. So you've got to start to deal with these threats. You've got to start to deal with them soon.
Preserving populations in the wild is vital, but is inadequate to alone protect our flora. We must respond through improving our off-site or ex-situ conservation methods, which is why the Germplasm guidelines have been updated.
There are new guidelines on the genetics of capturing and maintaining an ex-situ collection. There are also new guidelines for species that need extra care in ex-situ conservation, so orchids, special collections, and non-orthodox species as well. There's a lot of new information on identifying and conserving species that just don't suit conventional seed banking.
The updated Germplasm guidelines are designed with use in mind. Use by volunteers, researchers, students, community groups, not-for-profits and government. With over 68% of Australia's threatened plant species represented in conservation seed banks, the way we preserve populations off-site or ex-situ than ever. Ex-situ collections aim to safeguard species for use in the wild, either now or in a future we cannot predict.
Some very familiar faces and voices there. Well done again, Amelia. Well done, everyone. It gives me great pleasure to launch Plant Germplasm Conservation in Australia, Strategies and Guidelines for Developing, Managing and Utilising Ex-situ Collections, 3rd edition, timely, necessary and perfectly pitched. Brilliant job, well done. Thank you. I think it's time for applause and fireworks.
Okay, that's really great to see that that launched there. Hello, everyone. Good morning. If you're in Australia, could be other times of day elsewhere, of course. My name's David Bush. I'm the director of the Australian Tree Seed Centre, which is in Canberra. And I should start, of course, by acknowledging the country that we're on. It's the country of the Ngunnawal and Ngambri people. and we had a very nice welcome to country yesterday. It was good to see. And in this session on gene banking and seed banking, we're really going to be talking about collections of seeds and germplasm that go across very wide geographic areas, and it's probably a good idea to keep that in mind and think about all of those lands, those of First Nations people that those collections are drawn from. So that's a good way to start. Actually, it's going to be a pretty good session, I think. Looking at the abstracts here, we've got, of course, a sort of a double act keynote here this morning, and I'll introduce the keynote speakers just after this. But the talks that follow deal with a range of things. In the first bracket of talks, we've got the issues in dealing with difficult storage behaviours like recalcitrance of seed. There's a talk on rainforest seed. There's another one on actually difficult storage or difficult or problems, issues with some broadleaves and cryo storage, all that sort of thing. Then into the next session, we've got a couple of interesting talks on microscopy, one, and on computer vision or recognition of seeds there. Another one on cryo storage, seed quality. And then in the final bracket, we've got a sort of a diverse mix of talks, again, from some international speakers as well. So we're covering quite a lot of ground in this session. Those talks include through those sessions people from China, Indonesia, India, and the US. I might have even missed something there. But in the abstract to our keynote, we're going to be talking a little those that will deal with the Millennium Seed Bank, which of course it's mentioned there that 95 countries are involved in that. So very, very, very broad global outreach. We should probably get underway with those keynotes. Now, we're going to have two people speaking, and these are people who represent some of the biggest seed and germplasm collections in the world. The first one will be Sally Norton, but I'll introduce her second. We've got Dr. Eleanor Bremen from the Millennium Seed Bank of the Royal Botanic Gardens at Kew. She's a senior research leader for seed conservation there. Background in plant ecology, forestry and paleoecology, and she's been working in seed conservation for 7 years. The person who's going to lead off is Dr. Sally Norton, who, of course, is the chair of the organising committee of this conference and who's already spoken this morning. She's from the Australian Grains Gene Bank, which is one of the most diverse agricultural ex situ collections. So two quite different collections these scientists represent here. She's a specialist in wild crop relatives, but I think she's going to give us a general overview of some of the activities that have been going on at the Australian Grains Gene Bank. So I think without further ado, Sally's going to lead off, and then we'll go straight into the presentation from Eleanor after that.
Hi everyone, I'm Sally Norton. Been introduced quite nicely there in terms of the agricultural gene bank that I'm talking about today. I guess I'm talking about perspectives from the agricultural side, and we're looking principally at our general intro to our activities and how that's perhaps slightly different to the conservation and restoration seed banks that we find around Australia and around the world. I guess firstly though, I'd like to let you know that I'm coming to you from the lands of the Watchbollock, Wagaya, Jupagulk, Jardwa and Jarba peoples, and acknowledge the traditional owners of the lands of which we all meet from today, and pay my respects to their elders past, present and future. So I guess, what does the Australian Grains Gene Bank do? Our mandate isn't that much different to other seed banks or gene banks around the world. We're acquiring germplasm, we're conserving it, we're maintaining it. And one of our big areas of activity is that we are distributing to agricultural research and breeding programs. Being an Australian gene bank, our mandate is for the benefit of the Australian grains industry. However, we provide seed to researchers all around the world upon request. One thing that a lot of people are not aware about is that 95% of Australia's agricultural plants actually come from overseas. That means they're not naturally within Australia. And so that's really important because we have very tight biosecurity borders in Australia, making introduction of plant species and anything really very difficult and under stringent compliance, as it should be, so that we are protected from the pests, diseases, and everything else that aren't a concern for us in Australia but are overseas. So the Australian Grains Gene Bank was established in 2012 with a new facility built in Horsham and opened in February 2014. And this represents an amalgamation of all of the previous grain crop gene banks around Australia. And I think we have material from four gene banks covering tropical, temperate, pulses, oilseeds and cereals, and a range of other more minor crops. We operate in the public domain under the Plant Treaty for Plant Genetic Resources for Food and Agriculture. And we operate very much within the global network of agricultural and conservation seed banks or gene banks. And that's important because we're reliant on this network to import new agricultural biodiversity for use in breeding programs. And we have the national focus, so we're looking at cold, temperate, right through to tropical, subtropical environments. So although we're based in Horsham, we are actually covering off a very wide range of activities. So we also conserve cultivated species. So that's varieties that are grown in agricultural fields here in Australia, so elite modern varieties, through to traditional landrace varieties from typically developing countries. We have a lot of breeding lines, which are lines that are bred for evaluation in various research and breeding activities. And importantly, we have a lot of crop wild relatives. Mentioned before, I'm a specialist in crop wild relatives. That's my passion. We have so many wild relatives of crops here in Australia that are underrepresented anywhere around the world in any ex situ conservation gene bank. And after going up to the territory over the last couple of years, seeing a lot of our native wild relatives are subtropical, tropical. So a lot of the collecting that we do around the sort of the north of Australia, we're seeing more and more the threats of urban development, weeds, and just changing land use on those species. The Australian Grains Gene Bank, we pretty much are an orthodox seed centre. So we have minus 20 degree vaults here. We have 3 large walk-in vaults here with a capacity we have a capacity of about 200,000 accessions. We have 180,000 accessions at the moment. So a very big collection. It's very diverse. I mentioned before we are quite large and we cover temperate through to tropical. And as such, we have more than 2,000 different species, not counting the subspecies, from over 150 countries of origin. And what makes this challenging is that in Horsham here, we can't really grow out or maintain new seed stocks of the tropicals. So that's done in a suitable location. And you can see down bottom right there, a bit of a breakdown of our collection here. So we're about 50% winter cereals, which is wheat, barley, oats, rye, triticale. And then the rest is made up with tropical and temperate pulses and cereals. Really quite important that diversity is recognised. Moving on to our core functions, much less like most gene banks, acquisition is a very large part of what we do. We bring in around about 6,000 accessions a year. Around about 2,500 of that is through plant quarantine. And the picture of the pots in a glass house there is our new plant quarantine facility, which has just been finished and handed over. And with that new capacity, that increase will go up to about nearly 4,000 accessions a year coming in. We obviously collect crop wild relatives from around Australia. That central picture there is of Jimmy on the top of a big rock that took us 45 minutes to get up looking for a plant, and it wasn't there, but it was a great picture, great view. And then the crew obviously out there collecting in that year. We also have a lot of domestic donations, and that averages between about 500 and 1,000 lines per year for the last 5 years or so. But in the last 12 months, that's increased to about 10,000 lines of being donated material of developed breeding sets or mapping populations. So there's an increasing focus on that for the Australian grains community to support that. And a lot of those have been referenced genetically and phenotypically. So very valuable sets, but exponential growth of the collection will result. Storage and duplication, no different to any other gene bank or seed bank, and similar to Millennium Seed Bank. We bring material in to meet whatever project needs are there. It's data-based, it's cleaned, it's threshed, it's stored, goes into the foils, into our vaults. We also back up into a domestic facility in Tamworth in New South Wales, where eventually 100% of our collection will be backed up. And then we also back up into the Svalbard seed vault with a deposit, combined deposit with Australian Pastures Gene Bank planned for early next year. In terms of maintenance of our collection, we undertake viability testing, around about 6,000 accessions per year. About 4,000 of that is new entries, so baseline, and then about 2,500 of those would be periodic tests going back into the historical collection and doing a retest every 5, 10, 20 years, depending on the species. We do seed multiplication and seed regeneration here. So often coming out of quarantine, we only get a small amount of seed. So we have to go through an immediate seed multiplication, and that's undertaken in either pots or in the field, as you can see there in some of those pictures. And we also do seed regeneration, where we have seed accessions which have low viability or decreasing viability and low seed. And so each year we do a mixture of the multiplication and the regeneration. To maintain purity, that's a very important thing for us. Our regeneration and multiplication protocols are species specific, but everything is hand harvested, which is quite backbreaking in that. The main function of what we do in the service, the client service function we do, is the distribution. When we started the AGG in 2014, I think we sent out about 13,000 accessions, or 2014, I should say. We're now up to about 33,000 accessions per annum. During COVID, we had a near doubling 6-month period, which was really nice, but it really put us under pressure with COVID restrictions. As I mentioned before, we're primarily here to support the Australian grains industry, but about 10% of our distributions on average are going to international researchers. Some of that's repatriation, and some of that is for research use primarily, and some of that is for accessing some of our Australian native wild relatives which aren't represented anywhere else in any other gene bank around the world. And there is very much an increasing trend to crop wild relatives. Sorghum, pigeon pea, and rice are primarily the bigger ones, with a movement towards mung bean relatives as well and soybean relatives. We also have some diversity sets in here, which are increasing in use. So that's about 50% of our activities in terms of maintenance, and about probably another 30% with distribution. So they're our two main activities that we do. Of course, with every gene bank, there's the usual gene bank management, protocols, procedures, policies, standards, databasing, and of course, it's the relationships, the partnerships, and the collaboration, which is really important for us in running an effective gene bank here. And those linkages with either funding bodies or other seed banks and gene banks, or partnerships such as the MSB and the ASPB, have been fantastic for us to move forward. Another thing that we actually do, which is more on the fun side of science, is we do some outreach. So we have some education program aimed at 10 to 12-year-old kids called Get Into Grains, and that's learning about where their food comes from, biodiversity, why seeds are different, and they get to store some seeds forever in the freezer, which they all kind of love. We also have art linkages, and we do a lot of capacity building as well. So I guess a little bit more. I've got a couple of slides to go, running a little bit over. Research, we do active research here on seed storage and longevity, genetic diversity of subsets through partnerships and through one of our scientists here, Erica, using a range of technologies and range of crops. We heard yesterday from Solomon talking about our post-entry quarantine diagnostics. That's an ongoing research focus for us to get the most effective and efficient screening for pathogen diagnostics, which is mandated by the federal government for us to do through our post-entry quarantine. And of course, we're supporting PhD students. We have one at the moment looking at diversity of mung bean for suitability for growth in Victoria, not naturally grown out in here. One of the big things is capacity building that we do. We have done some nice programs in the last few years with partnerships with ASPB, MSPB and the Offord Fund, with crop wild relative collecting, with documentation, gene bank core activities and trainees coming from within Australia and also from overseas, from developing countries. So PNG, West Papua and Myanmar specifically in the last few years. One of the big future directions for us is the crop wild relatives. We have a little bit of an overlap with the conservation seed banks in this area. And you can see on these maps here on the left, you've got the distribution of crop wild relatives in Australia. So you can see how rich we are, particularly across that tropical monsoonal area of Northern Australia. And on the right, there's the gaps in the collections. And you can see there's not a huge amount of difference in those maps. So that's a massive focus for us moving forward and through linking in with various government organisations to get that done. Future directions also for us, we have challenges. The collection is growing faster than we can actually maintain this, and our resources are finite, which is no different to anywhere else. And I guess we're really pondering the question, do we really need to keep everything? We know we've got duplications, but what we don't know is what's the representative diversity within those collections. Are they actually the same or not? And how do we stay relevant to end users? For us, our main function is supporting the grains industry. So our end users are moving to much more high throughput technologies, and really the omics, so genomics, phenomics, any other omics we can think of, are really where we need to move. And that's the focus of what's going into our strategic plan over the next 10 years. And really the greater knowledge of seed lines that we have in the gene bank here will significantly improve germplasm use and breeding efficiency, and underpin food security in a far more effective way. So in closing, thank you to the ASSC for the opportunity to present, and also to my team, partners and collaborators who've worked so hard to conserve our germplasm to secure our future. Thank you.
I'd like to thank the meeting organisers for inviting me to speak to you about the Millennium Seed Bank Partnership and really talk a bit about seed banking from the conservation perspective. So I think many watching will be familiar with this building, which is the Millennium Seed Bank. It's based in West Sussex, which is where I'm now, down in the southeast of England. In 2000, with the capacity to bank collections from up to 75% of the world's flora that bears orthodox seeds, and 500, a very grand ambition. We're only just on our journey into using this facility. Tues recently published its manifesto for use for the next 10 years, and we can see here the mission is to understand and protect plants and fungi for the well-being of people and the future of all life on Earth. And to coincide with this, Tues also releasing its new science strategy later this month. And the Millennium Seed Bank Partnership is one of the key initiatives within that strategy. And we will continue to strengthen this global plant conservation network and deliver solutions to biodiversity loss. So I think there are two main things which make the Millennium Seed Bank Partnership really unique. And the first of those is the partnership itself. This map shows you its extent since 2000. In that time, we've worked with 97 countries and territories to help conserve their national floras, both in the country of origin and with duplicate collections sent to the Millennium Seed Bank in the UK. We currently have active agreements to those partners and funded programs in about as many countries, although obviously we're maintaining links and dialogues with many more partners at the same time. The other thing that makes the Millennium Seed Bank Partnership unique is that it is focused solely on wild plant diversity and its conservation. So the slide here shows some examples of things that are banked at the Millennium Seed Bank. And, you know, this really sets us apart from all of the agricultural related gene banks. Last year, we were lucky enough to celebrate our 20th anniversary, and we still managed to get together to celebrate, just not in person, but on social media. So we had a 24-hour party, and thanks to in the Southern Hemisphere who kicked that off for us so brilliantly. The event was picked up by many national and international news feeds, including this one, The Week Junior, which is a news magazine for children, which really took our message of what we're doing generation. My kids certainly enjoyed reading about it. And for those already in the partnership and policymakers, etc., we had a special edition of Samara, our international newsletter for the partnership, which looked back and reflected on some of our past achievements. So I thought if I just showed you some of the figures from that, it would bring you up to speed on what we've achieved to date. Since 2000, we've worked with over 260 different organisations to help bank national floras around the world. And through this, we've trained more than 2,000 people who often go on to manage their local collections or national seed banks. Across the globe, the partnership has banked over 230,000 accessions of more than 50,000 species. And at the Millennium Seed Bank alone, we hold 98,000 accessions of almost 40,000 species. So the seeds that we hold at the seed bank come from over 190 countries and all 7 continents. And they also represent material from all 36 terrestrial biodiversity hotspots. So I manage the Millennium Seed Bank Partnership team. And it's really the role of my team to develop new partnerships, maintain existing partnerships, and create project portfolios that are bringing the seeds into conservation, and also develop work packages around research that can further our understanding of those seeds and how we might best conserve them going forward. So it used to look more like the top picture there with people meeting in person, but as we all know, it's more like the bottom now. So I'm talking to you over Zoom currently. So all of our partners have an access and benefit sharing agreement with us. This sets out not only how we work together, but also what can and importantly cannot be done with any material that's transferred to Q, providing that prior informed consent. The main areas in which we work cover all aspects of seed conservation, so through from the planning in the collecting stage, and that includes gap analyses of what material still needs to be collected, right through processing, storing, bio data management and distribution, and seed bank management. So these standards form the basis of our training and technical support to partners, and we're continually striving to ensure that not only the Millennium Seed Bank, but also those in the partnership meet these standards. And this will mean that their collections are of the highest possible quality and therefore of the greatest possible use. One of our main areas of outreach to practitioners is through knowledge sharing, and we do this in a variety of ways. First of which is training courses. We hold them in country and at the MSB several times a year, and they're tailored towards the participants' needs. We can also provide information on seed bank design and equipment purchase and use, and we have expertise in data management associated with long-term seed collections, and we have created the data together information on the collections held across the partnership. So I think for us, this kind of knowledge sharing is one of the most important aspects of our work. An additional area of outreach is seed supply. You can go to this website here, Seed List, and request seeds from our collections. We've sent out material from about 12,000 collections so far. It's free of charge. You get about 50 seeds. Most to date have been used in research, in conservation, education, display, etc. And in addition, we have other ways in which our seeds are used. So some projects are specifically set up to enable seed use, such as the Crop Wild Relative Programme, where seeds were sent out to pre-breeders at the end of our collection, including some in Australia, so that they could be characterised and traits which might be useful for future crops could be identified. So final area of outreach that I wanted to mention is research. At any time we have a number of programs undertaking research both at Kew and across the partnership, and I've just highlighted two here on this slide. The first is through the Global Tree Seed Bank programme, which is conserving threatened trees across currently 8 countries. It's in its 3rd phase. And we've got one piece of work ongoing at Kew under this, which is improving the preservation of difficult to store tree seeds. So currently in the labs, we're looking at seed coat thickness and the impact that that might have on their preservation. Under the Threatened Biodiversity Hotspots programme, which is currently active in 7 countries, I just highlighted here some work that's happening in Indonesia in research. So you can see that the focus is on particular floras or particular groups of plants and better understanding them and their conservation. So really the theme that underlies all of our conservation work is seed biology and conservation, and increasingly how that material might be used and what that might mean for restoration and livelihood outcomes. So finally, I just wanted to mention a bit about our future plans. In our new science strategy, we're setting out our ambitions for the coming decade, and the Millennium Seed Bank Partnership will continue to be a focus for Kew. So we will continue to conserve orthodox seeds, but increasingly we're looking at those exceptional species that can't under traditional seed banking conditions, and what can we do to better improve outcomes for them. With the development of a Kew cryosphere, that's going to be a new area of understanding and advance. We also, in this UN decade on restoration, are turning our attentions in this direction. We want to increase the supply of native seeds for restoration, and also our understanding of the traits and characteristics of particular species, which might be important for restoration, particularly in reforestation as well. And finally, we will continue our work in training and technical support and growing this area to help support the partnership and wider, and linking with others who are also delivering training in this sphere. So I'd just like to thank you so much for your attention, and I would be very happy to take any questions during the Q&A. Thank you.
That we've just had there. Thanks both for doing those. We've got Sally here on stage to take some questions, and Emilia Martin, I know, has one, and since she's here, she can ask it personally.
Yeah, thanks, David. So, Sal, in the germplasm guidelines, it's really obvious that the native plant gene banks have the acquisition, the storage, the duplication, and the maintenance in hand. But I think we have a lot to learn from agricultural gene banks like the Australian Grains Gene Bank when it comes to regeneration and multiplication of collections. Yeah, how do you think we can learn from your experience in the costs and the time frame and the kind of skills that are necessary for that regeneration and multiplication part of your work?
Thanks, Amelia. I think that's a pretty big question in there. It's very much a learning curve. I mean, we've got such a diverse collection here that we don't know everything about every species. And we use species where we actually know enough about to actually successfully create seed or regenerate seed. We use that as a guide, as a start, and then we can tweak it as we go along. In terms of the conservation seed banks, I think, the ag gene banks do really well in some areas of what we do, but we're certainly learning a lot about the regeneration. And I think the most appropriate environment is the key thing. If you don't get the environment right, you're pretty much going around in circles, I think. The people are the important thing, I think. The regeneration multiplication is the most expensive part of what we do because it involves a lot of people. So getting the right people with the right skill sets, and not everyone needs to know everything, but if you don't have the people within the team itself, but it's knowing who you can draw that knowledge from, which is really important. But the resourcing for that is hard to get right because there is always so much more to do than what we can physically do. And I'm thinking it would be exactly the same in the conservation system as well. That we're all great at collecting seed and conserving it because it's at risk or threatened or nearly extinct. But having a small number of seeds where you don't have enough to actually do that underpinning biological research to figure it out is the hard bit.
Thanks for that. There's another question's come through for you, Sally, from Catherine Whitehouse. The question is, what do you consider to be the biggest challenge that we're facing as an agricultural gene bank compared to the other native and restoration seed banks within Australia?
I think the biggest challenge for us is gene banks and seed banks have been around for a long time. There's a lot of us around the world, and we do have a lot of synergies in what we do. But the breeding tools and technologies that are being used to develop new, more resilient crops to either sort of be more productive under changing seasonal conditions and also into the future with the really unknown conditions under climate change, is that the genetic and the phenotypic tools they're using, they've changed the baseline on what information they want to select seeds for use in their programs. So for us going out and doing standard characterization on morphological traits, which is the way it's always been done, it's important. It's really important for quality assurance and it's really important for end use, but we need to do more. People want to know genetic traits. They want to know genetic diversity. And that's a key thing for us moving forward is we're not funded to do that. We heard from Robert yesterday that these technologies are getting cheaper and they're relatively quick. How cheap and how quick really is quite a challenge for gene banks that have got finite resources. So it's really through those partnerships and linkages with organisations such as Robert and people like Robert that we can actually start getting some of this done.
Okay, thanks for that. We're going a bit over time, but there's just one last question from Damien Wrigley, and I'm just going to add a little bit on it myself. How do you prioritise the crop wild relatives that you're targeting in Australia, and what is it about Australia's crop wild relatives? Why are they so important? Are there particular traits, drought resistance, or something like that? What is it?
I guess so the global gap analysis on crop wild relatives was done by a range of organisations, including the Crop Trust, Biodiversity International, a little while back now, and they really did identify a range of global species that were really underrepresented across or any gene bank really. So we use that as a guide. So for us, the primary ones that are really underrepresented in the crop wild relatives would be the sorghum, the rice, the pigeon pea, the mung bean, the soybeans, the list goes on. There's a range of things there. So we basically prioritise based on those and we go in and target. So last season, April this year, we were lucky enough to actually get out of Victoria for 2 weeks. We actually went up and we targeted one sorghum species in particular because it's isolated in geographic distribution. So we targeted that. And then when we know we're going to that region, we then also map anything else that is on our target list that's in that area. So it's through linkages and partnerships where we can actually fluctuate with the target species of the year, but it's primarily based on the main crops that feed the world and looking at that. And you saw the list that Robert put up yesterday. That's far greater than what we've been targeting in the last few years. but it's further conversations with groups like Roberts that will allow us to target more over a wider diversity through networks as well.
Thanks very much for that. That's great responses. And thanks very much to you, Sally, and to Eleanor for those great presentations. Let you off the hook now as far as the questions go. I think we'd better get on with the next bracket of talks. We've got 4 talks before the break. Daniel, Karen, Xiaojian, and Bryn will be talking mainly about difficulties in seed storage and conservation. So we can start on that now. And people, please write questions in the discussion forum.
Due to the desiccation sensitivity of their seeds, it has been acknowledged that the long-term ex situ preservation of recalcitrant seeded species can only be accomplished by cryobiotechnology and the wide implementation of in vitro culture and cryopreservation methods. Excised seed embryos or embryonic axes are the preferable source of germplants for seeds of recalcitrant trees for 2 reasons. Firstly, because each embryonic axis corresponds to one seed, so their cryopreservation can capture a large genetic diversity compared to vegetatively grown tissues such as shoot tips. And secondly, because the germination of the embryonic axis and the production of a full plant can be started relatively easy in basic plant in vitro platforms. However, a relatively low success in the cryopreservation of embryonic axis for the temperate tree species studied up to date, often below 45% of plant in vitro regrowth, has hampered a wide implementation of embryonic axis cryopreservation of recalcitrant seeded species across gene banks. These results were us to study the factors that allow a better success in the cryopreservation of embryonic axis from temperate recalcitrant seeded species. We found, for example, that very fast drying rates, like those reached in the so-called flash drying methods, not only achieve higher plant growth rates, but also expand the water content range at which embryonic axis can be successfully cryopreserved. The water content threshold at which water freezing events are not longer detected by differential scanning calorimetry typically marks the water content limit for successful embryonic axis cryopreservation. In addition, we found that higher cooling rates tended to achieve higher plant growth rates at a global scale, although not for all the species when comparative studies were done in one single species. With the results of the survey in mind, we developed a cryopreservation method based on partial and fast drying and fast cooling of embryonic axis that were protected with antioxidants during the excision process. And we applied that method to the comparative studies in the Aesculus and Quercus genera, 2 of the most important recalcitrant seeded trees in Europe that are increasingly being included in global and regional IUCN red lists. We found quite a large variation in the response of the embryonic axis to cryo, with horse chestnuts being in general relatively more tolerant to cryo than oaks. Variation within oaks and horse chestnuts was also significant, and for example, high post-cryo recovery was found in Aesculus castanum and Aesculus californica, while Aesculus essamica and Aesculus indica showed the lowest post-cryo recovery rates. Independently of this variation in response, maximum growth after liquid nitrogen exposure was achieved after flash drying to water contents in the range and 0.38 grams of water per grams of dry weight for most taxa studied, with some exceptions like Quercus rubra that tolerated slightly lower water contents. The cooling rates studied show comparable results for embryo cryopreservation, but highest shoot recovery tended to occur after the faster cooling rate. And finally, When partial desiccation and fast cooling was not successful, we attempted to use diverse cryoprotectants. We found that while PBS2 resulted toxic for the embryonic axis of all Quercus species studied, glycerol increased root and shoot survival in Quercus robur embryonic axis. However, the increase in survival did not reach the high levels desired. We investigated the source of the response to cryopreservation within oaks and horse chestnuts. In oaks, we found that the Spanish species from drier habitats with warmer summers tended to survive cryopreservation better than those from wet and fresh habitats. However, in the case of horse chestnuts, we found that variation was better explained by the phylogeography, with species in the monophyletic Asian clade of Aesculus being more tolerant to the cryopreservation protocol used. Currently, we are studying the 3D microstructural characteristics of the embryonic axis to find if the tolerance to the stresses imposed by desiccation and freezing in the cryopreservation process can be explained at a global scale by anatomical and morphological features. Interestingly, in this project we found that pollen from all Quercus and Aesculus species studied was desiccation and freezing tolerant and was safely stored dry at sub-zero temperatures for at least 1 year. These results facilitate the creation of pollen banks for these recalcitrant seeded species, which could be a great complement to expand the genetic diversity of gene banks preserving embryonic axis or other tissues such as shoot tips or somatic embryos. One of the main conclusions from the research conducted in this project is that the technologies and methods to implement the ex situ preservation of many recalcitrant seeded species through embryonic axis and pollen cryopreservation are ready. We must encourage public bodies, botanical gardens, and gene banks with cryogenic capabilities to design implementation routines and start preserving these species with the methods available. Waiting for a highly successful and universal cryopreservation method for all plant species and propagules means delaying the preservation of threatened and endangered species and genotypes with recalcitrant seeds. We need to take action before it's too late and a large genetic diversity is lost forever. Thank you for attending this talk and to the organizers of this conference for allowing me to present my work. Special thanks to the students that perform most of these experiments in the lab and to the diverse institutions that have funded this research.
Hi everyone, my name's Karen Sommerville and I'd just like to acknowledge the traditional custodians and elders of Darkinjung country, the land I'm recording this video on. I lead the Rainforest Seed Conservation Project at the Australian Plant Bank, and today I'll be talking about our work to understand the storage behaviour of rainforest seeds. Australia has a variety of different rainforest types, and although those habitats cover less than 0.5% of the continent, they hold more than 3,500 different seed-bearing species. All of these habitats are under threat in one way or another, so we have a pressing need to try and conserve the species within them. Seed banking is the most effective option for doing that, but rainforests have a high proportion of species with seeds that don't tolerate drying and freezing. So we set out to see how many Australian rainforest species might actually be suitable for banking. We began by collecting mature fruit from 313 species along the east coast of New South Wales. The fruits were collected from cool temperate, warm temperate, subtropical and dry rainforest types and included fleshy, dry, dehiscent and indehiscent fruit types and seeds ranging in size from tiny specks. The seeds were extracted on arrival at Plant Bank and then their storage potential was assessed by testing how well the seeds germinated when fresh, after drying to equilibration with 15% relative humidity and after storage at -20 degrees Celsius for at least 1 month. We had germination greater than 50% for just 162 species. Of those, 22% were sensitive to drying and not suitable for seed banking, and the remainder were fully tolerant or at least partially tolerant of drying to low moisture content and potentially bankable. Of the species that were tolerant of drying, 107 were tested for tolerance of freezing, and around a quarter of those were found to be freezing sensitive or short-lived in storage at -20. Those species may still be bankable under the right conditions, so we're now running a project to identify more appropriate storage temperatures for them. It took a number of years to accumulate this data, partly because rainforest seeds can be difficult to collect in quantity, and partly because they can take a really long time to germinate fully. So to try and speed up this process, we looked at environmental plant and seed traits that could potentially be used as indicators of desiccation risk. We found that desiccation-sensitive seeds tended to be larger than tolerant seeds, and seeds with well-developed green embryos were more often sensitive than tolerant. We also found there were significant differences between desiccation-sensitive and tolerant seeds in terms of fresh seed moisture content, oven-dried weight, seed coat ratio, and PDS, which is a figure representing the likelihood of desiccation sensitivity calculated from seed dry weight and seed coat ratio. But even though the median values for sensitive and tolerant seeds differed significantly, there was a large overlap in ranges for each trait that made them difficult to use as individual predictors. So we then used ordinal logistic regression to model desiccation response against a range of environment, plant and seed traits. In the first model, we included data for 156 species and started with a full model, removing non-significant predictors one by one till only significant predictors were left. In this model, the only significant predictors were plant habit and fruit type, with a tree habit or fleshy fruit greatly increasing the likelihood of desiccation sensitivity compared to other habits. We then ran a second model incorporating significant predictors from the first model, along with fresh seed moisture content and PDS. This reduced the number of species to 75 by excluding small. With those smaller seeds excluded, seed moisture content and PDS were the only significant predictors of desiccation sensitivity. The results of these models were interesting, but weren't really useful for confidently predicting the desiccation response of an untested species. So we combined the seed trait data from this study with data from earlier studies on rainforest species from Australia, Panama, and China. We divided each data set into 4 to 6 categories and then looked at the relative proportions of desiccation sensitive and desiccation tolerant species in each. And this produced some really useful charts showing how the proportion of desiccation sensitivity increases with increasing seed moisture content and dry weight, and also showing a limit below which all or nearly all seeds were desiccation tolerant. The same pattern was found for PDS. So from these charts, we could see that seeds were most likely to be desiccation tolerant when fresh seed moisture content is below 20%, seed dry weight is below 20 milligrams, or PDS is below 0.01. Are most likely desiccation sensitive. PDS is greater than 0.5. So with this data, we thought a more useful approach to identifying the desiccation response might be to develop a step-by-step key. So we combined the most useful trends from this of several key earlier studies. The first was one that found that genus is a good predictor of storage behaviour if data is available for other species in that genus. The second was a study that found 99% of seeds with physical dormancy were also orthodox. The third was the original study showing that PDS greater than 0.5 was a good predictor of desiccation sensitivity. And the final one was a study that showed that green embryos were generally desiccation sensitive. With that data in mind, we developed this simple key to help predict the likely desiccation response of an untested rainforest species. We start by checking for information on other species in the genus. If no data are available, we go to step 2 and dissect the seed. If the seed coat is hard, we go to step 3. Test to see whether the seed coat is permeable to water. If the seed coat is permeable, we go to step 4 and determine oven dry weight and seed moisture content. If these data don't give the likely desiccation response, we go to step 5 and determine the seed coat ratio and PDS. And if still no joy, we go to step 6 and look at or green embryo. If that doesn't help, then we go to germination testing. We tested this key on the 156 species from this study, starting from step 2, assuming no prior data, and found that 141 species could be assigned to a desiccation response without germination testing, and 90% of those were assigned correctly. So we think this key will be a great aid to speed the conservation of rainforest species and will be especially useful in those cases where seeds are scarce or heavily dormant. So I'd just like to thank our sponsors for supporting this project, and thanks to you for listening. Bye for now.
Hello, I'm Xiao Jian from the Germplasm Bank of Wild Species, Kunming Institute of Botany, Chinese Academy of Sciences. The Germplasm Bank has stored seeds from more than 10,000 species. Among so many species, there are some species exhibited peculiar seed storage behavior. Today, I'm going to talk about the relationship between annular fatty acids formed TAG esters and annular storage behavior of Millennium species. Lipid is one of the main reservations of seed and also is the second most important edible calories resource for human beings. TAG is the main form of lipid reservation in seed. TAG in seed is usually composed of long-chain saturated or long-chain unsaturated fatty acids. There are also unique fatty acids in seeds of various plant species. Annular fatty acids can be very long to medium chain fatty acids or contain substitutions on the carbon chain or have variations in the type, amount, stereochemistry, and the position of unsaturation in the carbon chain. The aromatic and organic acids are very long chain fatty acids, contain hydroxyl groups. Kufia seeds are known to contain short to medium chain fatty acids. Some Kufia species, which have high concentrations of lauric acid and or myristic acid, may suffer intracellular structure perturbation caused by lipid crystallization at low temperatures and an immediate imbibition after cold storage. You can see the needle-shaped crystal in the cell of seeds that cold stored. This damage can be avoided by exposing seeds to a brief heat pulse of 45 degrees centigrade to melt solidified lipids prior to imbibition. The melt temperature can be inferred by a differential scanning calorimetry and can be predicted and explained by the relative fatty acid composition of the lipid. Millennium is a genus in the Euphorbiaceae or spurge family, comprising around 120 species. Most species are shrubs to small trees and are found in the Malay archipelago, but the genus ranges from Africa to Southeast Asia to Australia and the West Pacific. We found a similar response to cold storage and heat treatment after cold storage for some Millennium species. But unlike Kufia, 45 degrees centigrade did not work for some species like you can see Millennium paniculatus here. It needs at least 50 degrees centigrade to recover after cold storage, and optimally 55 degrees centigrade. The DSC traces peak temperatures are consistent with the lowest or optimal temperatures for heat treatment. The transmission electron microscopy showed that the lipid crystal is different from that of Kufia. It seems that the lipid body was simply frozen rather than form needle-shaped crystal. The DSC traces indicated that the embryo need a lower heat temperature than the endosperm. Also, for the tetrazoline test, after 45 degrees centigrade heat treatment, the embryos can be stained red, but not the endosperm. Also, you can see the TM also showed that the endosperm is still frozen, while the lipid body is relatively melt in the embryo. A 45 degrees centigrade heat treatment can melt the lipid body in the endosperm as well as in the embryo, and the lipid body recovered to the same level as the freshly matured seed before cold storage. We checked the fatty acid composition of Millennium species in the literature. Please notice that Terravia nudiflora is a synonym of Millennium nudiflorus. And we found two things. First, there is a very unique fatty acid called camelolanic acid. It is an octadecanoic acid with 3 conjugated double bonds and a hydroxyl group at the tip. Second, the 2 species have a very similar fatty acid composition, but very different DSC traces and the response to heat treatment. For Trevia nudiflora, 45 degrees centigrade heat treatment can melt the frozen lipid and recover the seed viability. But for Millennium philippiensis, which have a very similar fatty acid composition as the Trevia nudiflora, theoretically you can infer from the DSC traces it need a lowest heat treatment temperature about 60 degrees centigrade, but actually we have never managed to recover these species after cold storage. So the melt temperature cannot be predicted by the FV composition as in the case of Kufia. You can see instead of forming common TAG, where T means 3, containing 3 fatty acids, the hydroxyl fatty acids can form TAG esterides that contain more than 3 fatty acids. This is an example of TAG esterides containing 5 fatty acids, and also with a free hydroxyl group, which you can add another fatty acids. The Maudi Torf also indicated that the species Millennium philippiensis contain TAG esterides that are formed by up to 14 fatty acids, and the species, the seeds of Trevia nudiflora contain TAG esterides containing fatty acids up to, you can see here, 7 fatty acids. We also tested other species like Millennium abelta, Millennium paniculatus, and Millennium japonicus. We found that the species with a higher melting temperature or higher DSC peak temperature tend to have a stronger signal of TAG esterides rather than a TAG. Thus, in conclusion, the camelolanic acid in Millennium seeds can form TAG esterides, and lipid containing those TAG esterides would be frozen at -20 degrees centigrade. Melt the frozen lipid before incubation is essential for maintaining of the seed viability. Different species need different lowest and optimal heat treatments. Probably it is the composition of TAG esterides determine the melt temperature of frozen lipid instead of fatty acid composition. I would like to thank our team led by Professor Hugh Pritchard and Professor Yang Xiangyun. I also would like to thank Dr. Mark A. Smith and Dr. Yubu Zhu and Dr. Tian Bo for their help in this research. Thanks.
Hello, my name is Bryn Funnekotter. I'm a research scientist at Curtin University and King's Park Science in Perth, Western Australia, and welcome to my talk on cryopreserving recalcitrant seeded species. I'm going to talk quickly about our work on how oxidative stress has limited some of our cryogenic survival. So recalcitrant seeded species are unable to be dried to low moisture contents and are normally chilling sensitive. This makes these species unsuitable for traditional seed banking conservation. So these species tend to have quite large seeds that have very high water contents and are metabolically active, so they don't go into dormancy needed for traditional seed banking. So cryopreservation provides a long-term conservation tool for these kind of tissues. So it is the storage of material at very low temperatures, typically in liquid nitrogen, and these low temperatures help stop any of the biophysical biochemical reactions, basically keeping the tissue in stasis. So it is a very long-term storage option. There's many species that have been rewarmed from cryostorage after 25 years and shown very good viability. Additionally, it's applicable to a wide range of germplasm, so a lot of species can be conserved using this method. This method is a multi-stage method to get the samples into cryo storage and can require quite a lot of optimization for some species, from excision of the plant material out of the mother plant, desiccation to remove excessive water. A cryopreservation treatment is quite often applied to help limit ice formation. It is then cooled and rewarmed and recovered when needed. So we worked on 2 Syzygium species, Syzygium australe and Syzygium paniculatum. These species represent a sort of model recalcitrant rainforest species here that is accessible to us in Perth. So I've got some of the Syzygium australe growing in my backyard, which is very convenient for collecting samples. So we could collect and sterilize the seeds and store them for about 2 months at 4 degrees before we started to see oxidative damage. Quite often these species will also show a lot of fungal growth and bacterial growth that would limit the ability of us to maintain long-term seed collections for testing in cryopreservation. So we initiated these species into tissue culture as well to provide a continuous supply of germplasm in the form of shoot tips that we can use to help optimize the cryopreservation process. In addition, shoot tips are advantageous over seed axes as they are a bit more uniform in size, which makes developing cryopreservation procedures a little more easier. So we've seen a lot of what looks like oxidative damage during the various stages of cryopreservation. So the shoot, they won't grow or will callus up a little bit. So we started to alleviate some of this oxidative stress. So you can see a lot of these shoot tips, well, they might have started growing, they've completely blackened and will stop growing from this point in time. So the first step was testing the addition of antioxidants. So we used 2 antioxidants, glutathione and ascorbic acid. These are 2 of the main antioxidants found in cells. And testing these during the desiccation period, which is just before cryopreservation, showed that glutathione did have a positive effect for increasing regeneration after desiccation compared to the control, but the addition of ascorbic acid was probably a little excessive and probably dropped the pH of the media too much, affecting the growth of these plants. So we looked at the formation of 2 lipid peroxidation byproducts, MDA and 4-HNE. Both of these are quite toxic to cells and can cause mutations and other stresses. So, well, the addition of ascorbic acid was not beneficial. It definitely reduced the amount of lipid peroxidation seen. So fresh shoot tips have very little formation of either of these 2 byproducts, lipid peroxidation byproducts. The control which is without any addition, did show an increase from the fresh tissue, so we are stressing them. Oxidation is happening. It was interesting that glutathione did not seem to protect these shoot tips and seed axes from the formation of lipid peroxidation byproducts. So the desiccation step seemed to be one of the main causes of stress, and this is prior to we even get to any cryopreservation, so there's a lot of work to be done in optimizing this. You can see as we increase the amount of sucrose used in the pre-culture treatments from 0.2 molar up to 0.6 molar, we're losing about half of our shoot tips just to desiccation stress. So the addition of lower levels of ascorbic acid did show a slight benefit, and again, glutathione shows some benefit, but it's not a huge amount anymore, especially when we're using the higher pre-culture media. So after this point in time, we assessed the total antioxidant capacity of the shoot tips at these different stages of the cryopreservation procedure. At the start, during excision, there's quite a lot of total antioxidant capacity in the shoot tips, but after 2 days in pre-culture, we've already lost a significant portion of the antioxidant capacity. And the addition of the antioxidants is not particularly making a significant difference once in the cryoprotection phase. And once these samples have been cryopreserved, there's almost no antioxidant capacity left to recover from any additional reactive oxygen species that could be formed during the recovery phase. And this is likely leading to a lack of cryogenic survival. So we've been testing more recently a new combination antioxidant solution. So this is combining citric acid and ascorbic acid. This provides a bit of a better buffered solution at a higher pH, much of a pH stress as ascorbic acid on its own. And this is shown to be quite beneficial. So the samples are excised, in this case, Q-tips are excised from the tissue culture material, washed in the antioxidant solution for an hour, and then going through the cryopreservation procedure. And once we have the trees, again, we'll be testing this on the embryonic axes to see if it is beneficial for them too. So thank you to Robin, Hamish, Leigh, and Kalinda, who are honour students that contributed to this work, and my supervisors, Ricardo Mancera and Eric Bunn, and thank you for your attention.
Thanks for those really good talks there. We've got three of the speakers here on stage with us, and Bryn has got a question for Xiaojian. So we don't have much time, but please ask away, Bryn.
Thank you. So I was going to ask if the results you saw with the lipids freezing and rewarming, if that was exclusive to seeds with high oil contents, or is this sort of seen in seeds with lower oil contents as well?
Thank you. I think it's only for the oily seeds, because you have to have a lot of lipids to be crystallized, and if the seeds are not so oily, it won't be have this kind of phenomenon. Thank you.
Thanks very much. There's another question from Lydia Guya, which has come online for you again, Dr. Hu. Do you suspect any other genera might have similar storage physiology, or is it unique to the Molottus, the lipid problem?
Yes, actually there are some examples reported by Dr. Groot, I think, from Wageningen University, about the Chinese beta guard, from the Cucurbitaceae. Also, I think if you search the Euphorbiaceae family, I think there should be some other family genus will have the similar phenomenon, like Macaranga. Maybe I'm going to check that. Thank you.
Thanks very much. This is a quick general question for you, Karen. There's been a lot of worry here in Australia about megafires burning into the rainforest. Can you give us maybe something like a traffic light, red, amber, or green? What is the status, in your view, of ex situ preservation of our overall rainforest flora?
At the moment, we have very little stored in the seed bank, and it's something that we've been working on for the last decade to try and figure out which species we could actually put into the seed bank and which ones weren't suitable. But I think with the work that we've done, we've found out that quite a few species can be banked. It's just we need to go out there and collect them, and rainforest species are a little bit notorious for not producing seed when you want them to in the quantities that you'd like. So it's they take a bit more work than orthodox species, but we are definitely at a position where we can go out and collect and start banking more seeds.
Right. Okay. Yep. Well, thanks again for those presentations. Do we have any other further questions? I'll just check here on the discussion forum live Q&A. Can't see anything there. No, I think that's good, and that's been a good session. We've got a break now for 10 minutes until 11. So thanks again to all the speakers. I've really enjoyed it, and come back in about 10 minutes, please, and we'll keep going from there. Thanks, guys.